Well, without further ado, Island,
Yes.
well, first no, it's not a question, just hush. First, I got associated with Island as part of the collaboration for the LEAF project, and she was in another group, let's just say. And then she met the people that were working on LEAF in computer science and engineering and figured out that they were the cool kids. So she came over from the dark side and is now in two of my groups. Decided along with LEAF that she was going to see some interest, or that she saw some or had some interest in the Voynich manuscript, which is that medieval thing. And so has jumped into that full speed ahead and is bringing her unique perspective to it and really making as large a contribution there as she did in LEAF. So have I made up for all the bad things I do?
Just like we practice, Mr. Downing.
Okay.
That sounds wonderful. I'm very excited to hear what's going on here.
It's very good.
Okay.
All right, take it away, Island.
All righty. So, hi guys, my name is Island Salaifar. I'm a senior from Carmel High School, and today I'll be presenting an assessment of the heavy metal pollution, soil health, and potential remediation strategies for the Fremont Leaf Community Garden. During this presentation, I'll be covering a quick introduction of the Leaf Community Garden and the challenges it's facing, a look into the historical use of the site, and an overview of our heavy metal pollution and soil biological health analyses. Finally, I'll discuss suggested remediation strategies for the garden, key takeaways, and acknowledgements. To begin, the Leaf Garden is a nonprofit organization in Fremont, serving Fremont, Newark, Union City, Sunol, and Hayward. Their mission is to take positive climate action by educating the community to live sustainably, grow healthy food, and reduce waste. They achieve this goal primarily through their two cultivated gardens, the CR Garden and the Bee Sanctuary Garden, which have been cultivated for 7 and 2 years respectively. The harvest from Leaf's gardens feed people in need through donations to the local food banks. Leaf's third garden, known as the Urban Garden, is an undeveloped plot of land that the garden is currently working to cultivate. After development, the Urban Garden is slated to double Fremont Leaf's donation capacity to local food banks. Before this can be achieved, however, the garden faces the challenge of assessing the levels of heavy metal pollution present in the soil and analyzing how this will impact the biological health of the soil and consequently the crops grown in the plot. That's where we, the ASDRP LEAF team, come in. The problem of heavy metal contamination impacting the health of crops grown in the polluted soil that we aim to address with our research is not isolated to Fremont Leaf's gardens. In fact, widespread pollution of soils by heavy metals is a major environmental challenge throughout the world. According to the United Nations Environmental Program, over 20 million hectares of land are contaminated by heavy metals. In order to gain insight into the specifics of the urban garden plot to better understand the heavy metal contaminants we may be dealing with, we began our research by investigating the historical use of the site. The western portion of the Niles Canyon area in which the Leaf Stone Garden is located is known for historical presence of railroads. The urban garden is in fact bordered by a now unused railroad track. In addition to these railroads, which are immense sources of potential heavy metal pollutants, the property also has a long history of intensive mining and manufacturing uses. In 1907, a clay mine and brick manufacturing plant were built near the plot of land to manufacture bricks for rebuilding San Francisco and other parts of the Bay Area after the 1906 earthquake. The California Pressed Brick Company alone constructed 14 kilns, as you can see in this image on the right. From 1910 to the 1930s, the kilns and other facilities used a variety of heavy metals and oils to fuel manufacturing, all of which left a significant impact on soil. In addition, there's currently a plating industrial plant adjacent to this plot of land, which may also be a source of the heavy metal pollution. With this understanding, we began our analysis of the heavy metal pollution in the urban garden soils. Our methodology consisted of sample collection from 3 distinct sites around the garden, one site adjacent to the fence separating the railroad track from the urban garden, one site in the middle of the garden, and the final site near the entrance to the garden. In order to produce a baseline or control set of data to compare our urban garden samples to, we also collected data in triplicate from the local ecology, for which we chose the undeveloped sites along Mill Creek Road in Fremont. These samples were then analyzed for the presence of heavy metals using the technique known as X-ray fluorescence, or XRF. XRF is an instrument that determines the chemistry of a sample by measuring the fluorescent or secondary X-rays emitted from a sample when it is excited by a primary X-ray source. Each of the elements present in a sample produces a set of characteristic fluorescent X-rays, like a fingerprint, and that is unique for that specific element. This can be used to identify which heavy metals are present in a soil sample. Analysis of the urban garden data that we collected in comparison to our local ecology samples, as well as FDA-approved limits for heavy metals in agricultural soils, indicated our 3 primary elements of concern to be cadmium, nickel, and zinc. We also mapped our element of concern of cadmium to that plating industrial plant that I mentioned before. After comprehending the primary heavy metal contaminants present in the urban garden soils, we then set out to understand how the presence of these contaminants impacted the biological health of the soil and subsequently the crops grown in the soil, which families across 4 counties currently depend on for their fresh produce. In order to most directly assess the impact of the soil heavy metal pollution on the health of future potential urban garden crops, we collected more samples from the 3 previously determined test sites and planted Buttercrunch lettuce plants in the soil in 3 separate pots. For the control group, we utilized purchased potting soil in order to have a baseline of crops grown in entirely heavy metal-free soil. We chose Buttercrunch lettuce plants due to their sensitivity to being grown in polluted soils. We chose these plants specifically to observe the impact of the urban garden's heavy metal pollution on a more susceptible crop, rather than one that was more hardy or resistant to contamination. Plants were grown in a grow room with conditions controlled for light exposure, temperature, and water in order to ensure that the soil was the only variable acting on the plant's health. In order to directly assess the health of the Buttercrunch lettuce, we looked into the soil bacterial profile, the chlorophyll content of the lettuce grown in the soil, and the average soil pH weekly for 3 weeks after the plants had reached maturity. We found that the majority of these values fell well within the natural or accepted ranges for agricultural soil. However, interestingly, the plants grown in the soil taken from the test site nearest to the fence separating the urban garden from the railroad track demonstrated values toward the lower end of the healthy spectrum across all 3 tests, indicating that the nearer to the railroad track, the more pollution was observed in the soil samples. Finally, with a full understanding of the heavy metal contamination of the urban garden and its impact on future crops, we began looking into various remediation pathways toward improving the state of the soil prior to cultivation. First, we looked into in situ remediation, which is a process that refers to the cleanup of contaminated soils without excavating or removing contaminated media from the subsurface. Among the in situ remediation options we investigated are bioremediation through biochar application and phytoextraction. In situ bioremediation is defined as the use of biological processes to degrade, break down, transform, and remove contaminants from soil and sediments. Bioremediation is an effective means of mitigating petroleum, hydrocarbons, and metals, and also causes minimal disturbance to the environment at the contaminated site. Phytoextraction, on the other hand, is an in situ technique that uses plants, some specific more hardy plants, plants to absorb large amounts of heavy metals from contaminated soils and sediments for storage in their roots and shoots. This process involves cultivating plants in the contaminated area. We also investigated ex situ remediation avenues, which refers to the removal of contaminated media, either for off-site disposal or for on-site treatment and subsequent return to the subsurface. Our options included electrochemical remediation and land farming remediation. Electrochemical remediation technologies use AC or DC current in the treatment process to either mobilize or break down contaminants in soil. AC-DC converters produce a low voltage electrical field that polarizes the soils, causing redox reactions that mineralize organics and increase the mobilization of metal. Metals then migrate to the electrodes where they are deposited and removed. Land farming, on the other hand, is a medium to long-term remediation technology that successfully treats contaminants through frequent tilling. This technique is most effective for oils and pesticides, which are two other contaminants which we suspect to be in the urban garden soils due to the historical tracing we did and investigations into the plants that are currently surrounding the plot of land. However, we have not had the opportunity to fully research those other two contaminants. Finally, we conducted a cost analysis of the different remediation strategies and found bioremediation and phytoextraction to be the most cost-effective. So, some key takeaways. Based on our remediation strategy analysis, our final recommendation involves combining plants and microbes for the remediation of the urban garden's heavy metal polluted soils. The combined use of both microorganisms and plants for the remediation of polluted soils has shown to result in a faster, more efficient cleanup of polluted sites. The success of the combined use of these organisms depends, of course, on the species of microbes and plants involved, and to some extent on the concentration of the heavy metals in the soil. We plan to investigate both of these remediation techniques in the future. Regardless of which remediation path the leaf garden decides to select, the criticality of maintaining the health of our soils remains evident. Soil is the base for terrestrial life, and by treating it as a sink for our pollutants, we will only end up poisoning not only ourselves, but all aspects of the environment as well. Through careful lands and waste management, we can prevent the need for such soil contamination assessments and remediation attempts, such as we are trying to do in this study. Finally, acknowledgements. I'd like to thank my advisor, Mr. Downing, for that wonderful introduction before this presentation, as well as all his mentorship throughout the semesters that I have been in his group. I'd also like to thank all the team members with whom I've had the privilege of working. I'd also like to thank the Fremont Leaf Garden for allowing us to conduct this research on their land, as well as ASDRP and the Olive Children's Foundation for providing laboratory space, resources, and research opportunities to high school students. Thank you very much for listening, and I can take any questions now.
You see the one in the chat?
Oh, yeah. So Ananya asked, I don't know if I missed this, but why did you choose Buttercrunch lettuce specifically? That's an excellent question, Ananya, and there are several answers. I'll start with the simplest. First of all, we wanted to conduct this study preferably in less than a 2-month to 3-month window, and Buttercrunch lettuce grows very, very fast. It reaches maturity in about just a month. So after it reached maturity, we were able to conduct our tests in 3-week intervals, and we were able to be done with that study within 2 months. So that was one reason we chose Buttercrunch lettuce. The next reason is that, actually there are 3 reasons. The second reason is that Buttercrunch lettuce is a very sensitive plant to being grown in heavy metal contaminated soils. We specifically wanted to test a more sensitive plant because that way we'd be getting an example, or we'd be getting insight into what it would look like if the leaf garden decided to grow a crop that was similarly sensitive, what the impact would be on the health of that crop, rather than if we had chosen a more hardy or resistant crop to heavy metal pollution. Yeah, and the third reason is that the leaf garden had provided us with some insight into what plants they were looking to plant first, to cultivate first, and they said that they were leafy greens, so Buttercrunch lettuce fulfilled all of those requirements. So Connie's asking, does phytoremediation harm plants? That's another great question. So in phytoremediation, you are looking to specifically use some more hardy plants, like I was discussing in the previous question, such as eucalyptus, to remove heavy metals from the soil. And essentially, the plant is able to store, the most hardy ones are able to store up to 1,000 times more heavy metals in their roots and their shoots, as opposed to more sensitive plants like Buttercrunch lettuce. Now, this, of course, is not a great thing for the plant in question that's phytoremediating the soil, but it allows the soil to be cleansed of those heavy metals, and then other crops can be planted after the phytoremediants are removed. Usually phytoremediants, especially if they are being used in a heavily contaminated site, they'll actually be treated as waste after they're removed from the soil. So it gives you an idea. If the plants are being treated as toxic waste, then you get an idea of how great of a situation it is for those phytoremediant plants. So it does harm those plants, but that's the purpose. They're basically like sponges, sucking up all of the heavy metal contamination from the soil. Isaac Lee asked, for electrochemical remediation, is the current enough to pull the metals through the soil, or do you need to add a fluid to the soil? That's a great question. So for electrochemical remediation, there are several different types, and it really depends on the concentration of the heavy metals in the soils, the land area that or the cubic meters of soil that you're attempting to remediate, as well as which specific heavy metals you're looking to remediate. So it really depends on all three of those factors. And we didn't look very extensively into all of the electrochemical remediation methods, but if that's something you're interested in, it's a super cool remediation technique. I'd really recommend looking into it. Shreya asked, does it affect fruit/root-based beet/potatoes or just plants like lettuce? So I'm assuming you're asking, does heavy metal contamination in the soils impact fruits and root-based plants? Wonderful question. Absolutely. Heavy metal, so like I said, soil is the basis for all terrestrial life, essentially. And plants, through their xylem and phloem tissues, they get all of their nutrients and everything they need to survive, except obviously for sunlight from the soil. So when the soil contains contaminants like heavy metals, it degrades many of the plant systems and structures, and it causes damage. You asked about root-based plants. Those specifically will be even more impacted. And a lot of phytoremediants are actually root-based plants because they are directly sucking up those heavy metals from the soil. So yeah, it affects all plants. No plants are immune to being grown in heavy metal polluted soil. Some are just more resistant. Awesome. I think those are all the questions in the chat.
Okay, wonderful work.
Yes,
So,
very nice.
yes, wonderful. So couple of questions. I think you go with the chat one first. I'll go last.
Vibehub, I'm sorry if I mispronounced your name, is asking, apart from the methods that can be used directly to solve the problem, do you think that you could use these solutions and go further to even prevent heavy metals leaking into the soil? So you're getting right into the heart of the issue, and I love that. You know, that question gets into a lot of systems because particularly in America, we've had a culture of treating the environment like our garbage can for such a long time that majority of our soils are already contaminated. In fact, I think there's a statistic from the Intergovernmental Panel on Climate Change that says something like 97% of the world's soils have already been in some way contaminated or degraded, not all to the same degree, but in some way impacted by human influence or global climate change already. So it's a tricky question to ask, well, can we prevent the heavy metals from being introduced into the soil in the first place? Because they're already there. But can we do things to prevent those heavy metals from the heavy metal levels from increasing? Absolutely. And that gets into, a combination of policy, incentivizing corporations to dispose of their heavy metal contaminants through the proper channels. And, just in general, creating a culture of treating our environment with more respect and, more care, because it's already a big problem, but it's going to be an even bigger problem soon if we don't start taking it seriously. Awesome.
Okay, so I have two questions. First, let's go with, I think, slide number 17. So you mentioned about cost of each method. Define cost, like, what do you mean by cost?
Yeah.
Is it just the method? Is it method plus the labor? Is it method plus the labor plus time? Because sometimes the cost based on money is low, but when you look at the time frame, is taking so long that is not feasible,
Yeah.
So let's talk about what do you mean by cost?
Yeah, so for this cost analysis, we basically looked at the bare minimum because, like you said, based on the labor, based on the time frame, based on the area of soil that you're trying to remediate, based on the level of heavy metal contamination, all of those costs could vary so much. So we were looking at baseline, how much does this technique cost per ton of soil? We didn't look into labor or the time frame because, like, for example, with the leaf garden, there's so many other factors involved. The urban garden plot is actually leased to the leaf garden from ACWD because there is a water reserve underneath the plot. So there are all these complicated factors involved that could influence the time frame. So for the cost analysis, we just looked at the raw technique. How much would that cost per ton of soil? Okay, so there is a chance that there might be a chance that even the most expensive one will be the efficient one based on time and labor, probably. Yeah, absolutely, yeah.
Okay, so the second question is that what I understood that, correct me if I'm wrong, that this study is entirely on the soil base, right?
Yeah, so the soil base, and we looked at plants grown in the soil.
Okay, and you use the plasma emission spectroscopy for the plants, correct? So you use the spectroscopy to check the heavy metal, right?
Yeah, we use X-ray spectroscopy, but yeah.
Okay, and then was it? Correct me if I'm wrong. Was it the same thing between the plants and the soil in each site 1, 2, 3, 4, 5, if I'm not wrong, right? So was it highly correlated, like the plant which the heavy metal was more detected in site 3, for example? Was it the same thing with the plant, or?
Yeah, that's a great question. Actually, I'll share my screen again because one of my images shows this really well.
And it was just for one plant, right? Only one kind of plant for the butter lettuce, right?
Only one kind of plant.
Okay.
And actually, can you see my mouse?
Yeah.
So if you look right here, this was at the beginning of when we started doing our testing, once the plants had reached maturity. And if you look right here in the middle, this plant, I mean, you can just tell visually that it didn't reach the same height or width as the other plants did.
Mm-hmm.
This plant, the soil that it was grown in was the soil taken from the test site, not the test site, the sample collection site that separated the garden from a railroad track.
Activities and awards on
Sorry, someone talk. Oh, I think someone's just not unmuted. Yeah, so does that answer your question? Did I understand correctly?
Yes. But yeah, I mean, I'm just curious, how it will go with different plants, and it's just is it possible that specific plants, filter out not getting the heavy metals? I'm just it's just curiosity, just lots of questions in my head.
Yeah, absolutely. So that's right on the money. every single plant will have a different response to being grown in heavy metal contaminated soils. You know,
Okay.
Buttercrunch lettuce are much more sensitive plants, but for example, eucalyptus, duckweed, cattail, these are all very, very hardy, heavy metal resistant plants that are able to survive and even thrive in heavy metal contaminated soils. It really depends on concentration, type of heavy metal, soil composition, so many factors involved.
Yeah, I don't have knowledge much in botany, but I think the roots of the plants also has an important factor on filtering out the heavy metals to getting into the plant. So the root analysis of plants maybe will be an interesting part of, extended work too.
Yeah, absolutely agree. I have quite a bit of experience working with rhizobacteria with a partner. We did a project, and rhizobacteria are the trillions of microbes that inhabit the soil surrounding plants' roots. And essentially, they're exchanging compounds and nutrients with plants in this really complex symbiotic dance. That rhymes. I don't know why I said that, but it rhymes. And when they're doing this, they're able to support plants that are being grown in heavy metal contaminated soils, highly saline soils, soils that are heat stressed. So a bunch of these factors, the length of the roots, the width of the roots, how much rhizobacteria there is present around the roots, all of these factors influence the plant's uptake of those heavy metals and its ability to respond.
Wonderful. So Eileen, your work is great, but I would like to add that you are a wonderful presenter. I was just like enjoying, and I just wanted to you present and talk. The knowledge is flowing smoothly, and it's just like so pleasant listening to you. It was wonderful. Good job.
Thank you so much, Ms. Jahanika. I really appreciate that.